This call is now being recorded. Can you see the screen, children?
Yes, ma'am.
Yes, ma'am.
Yeah. You may not listen, but the titles are there. So you observe the titles, read the titles carefully. I'll make the entire screen full screen. RNA polymerase attaches to gene. See how the RNA strand is formed. Complementary bases are added. This is called transcription. Then this messenger RNA needs to be present. Non-coding sections are removed. It moves out of the nucleus and comes in the cytoplasm. This is now mature RNA. Ribosomes, smaller and larger subunit. First attaches. This is today's part, translation. Listen up. Three bases at a time. How tRNA is added, and each carries the amino acids. So how these amino acids are attached. This is a chain of amino acids. See how it is formed. Then this chain of amino acid forms a 3D shape structure that is called as protein. Okay, have you understood up to this? It shows both the parts, right? Yes, yes. Now we will start with the next part. I'll explain it first. Now, can you see the screen? Yes.
Yes, ma'am.
Ah,
Yes, ma'am.
so yeah, so that now, and the next part of this protein synthesis is called as translation. We discussed transcription in the last lecture. Now we will see what is translation. And again, if we get the time, then one more video in detail about the translation I will show you. Okay. So before we come to this part of translation, you should know the structure of mRNA and tRNA in detail. So I will revise it with the help of these diagrams. Now, can you see the arrow which I am showing here? This is, can you see the arrow? Just let me know.
Yes, ma'am.
Ah, so this is the structure of mRNA. It is labeled also here. mRNA. How it is? It is simple single-stranded structure on which these nucleotides, that is, these are arranged in a sequential manner, and this single stranded structure of mRNA is having 5 prime end, where I am showing you here. This is the left hand side, 5 prime end, and the other one is called as 3 prime end. That means 5 prime end and 3 prime ends are present to mRNA. Now on this you can see these G, U, C, all these nucleotides, right? A group of 3 is considered to be a codon, which represents a amino acid later in the later stage. So a group of 3 nucleotide, it represents a single codon. So in this way, codons are present on the mRNA. So such type of codons. Now GUC, this is first codon, CAG, second codon, GAG, third codon, CUA, next codon, in this way. Okay. And always the first codon is AUG generally on the mRNA when it is taking part in the process of protein synthesis or such reactions. So the first codon is called as start codon or initiation codon. Start, which is going to start the reaction. So this is called a start codon or initiation codon. Whereas the last codon is called as stop codon, which stop the reaction of protein synthesis. So this is called a stop codon or termination codon because it terminates the reaction. That is why this is called as stop codon or termination codon. And one more name is there, as it do not sense any of the nucleotides or amino acid or the meaning. That is why this is also called as non-sense codon. So it doesn't give the sense or it doesn't show or represent or codes for anything. So that's why this is called as nonsense codon. So first codon, which initiates the process of protein synthesis, is called as start or initiation codon, and the last codon in the synthesis is called as, which terminates the reaction of protein synthesis, is called as termination codon or stop codon. Now, up to this structure of mRNA is clear, I think. Now, the structure of, sorry, this is mRNA. I told you now about the structure of tRNA, which you have already studied last year, but still, you should know all the basic things which are very important in case of tRNA. Now, you can see tRNA is with these 3 arms and loops, and one is free without any loop, and these 2 arms are unequal. So this shorter one, which ends with 5 prime end, and the longer one, it ends with 3 prime end, and this longer one, longer arm, which is free, that is 3 prime end, that arm is called, or that site is called amino acid acceptor or attachment site. This 3 prime end of tRNA is called as AA site. That means amino acid attachment site. Now, one more thing is very important to remember in the structure of tRNA is that opposite to these free arms, whatever the end is there, that means this arm which contains the loop, this is called as anticodon loop. The word anticodon, why? It is very easy to remember. Anti is against, opposite, like that. Codon, that means the nucleotides which are present on this loop, they are complementary. Opposite or against or means what? That is complementary to the codons on mRNA. So the word is anticodon, and this is having very, very important role in the process of protein synthesis. So tRNA is playing very important role along with mRNA in the process of protein synthesis. Up to last part, that means transcription part, we studied mRNA, which is produced in the nucleus. And then after the processing, it comes to the cytoplasm where it is required. That means ribosome. That is what the site of protein synthesis. Now, same way, this tRNA is also playing very, very important role because it has these two important points. That means the free 3' end is useful for the attachment of amino acid required for the protein synthesis, and this anticodon loop, which is opposite to this free ends, free arms, that represents, that means the nucleotides which are present on the anticodon loop, they are complementary to the codons on mRNA. So this is very important. So remaining structure, I will not explain right now about tRNA, but these two important points you should remember and understand because it comes in the next part, that is the actual part of protein synthesis. Now, why this is considered to be the adapter molecule, because this amino acid, proteins are made up of amino acids. So DNA only decides which type of protein is to be synthesized. So accordingly, the type of amino acids, the number of amino acids, the sequence of amino acids, that represents the type of protein. So these amino acids are very important, but they are not in a position to directly come in contact and play the role in the protein synthesis. So that adapter is required, and that adapter is what? TNA. That means this tRNA is used as an adapter. That's why this is carrying, tRNA is carrying these amino acids. Now, one more thing, that last time also I told you during the DNA replication as well as in the synthesis of mRNA, that whatever the nucleotides are required, these nucleotides should be charged. Then only they will be playing the role properly. Like everyone needs energy, and that we take from food, the same way that energy is taken from ATP for the activation, right? For the activation, this energy is taken from ATP. The same way here also, these amino acids also need to be charged, right? And these are also needed to be activated. So what is the next part? Now we will talk directly about the process. Once now you have understood about the structure of mRNA and tRNA, now we'll move for the actual process of translation. Now what happens? See, translation is the mechanism in which codons of mRNA are translated and specific amino acids in a sequence form a polypeptide on ribosomes. Yesterday only I told you what is that polypeptide. Poly means many, peptide bonds are formed where between the amino acids, then only it will form a chain of amino acids, and that is nothing but protein. And this is what transcription we discussed. That means in the process of protein synthesis, first step is transcription. That means the information required for the protein synthesis is taken, transcribed on mRNA in a code language. Whereas in the next step, translation from mRNA, that code language is to be translated, that to form proteins. So this is what the second step now we are talking about. The process of translation requires amino acids, mRNA, tRNA, ribosomes, ATP, MGINs, enzymes, elongation, translocation, and release factors. Now just now I explained you what is that amino acid, and proteins are nothing but a chain of amino acids, that is called as polypeptide chain. mRNA, just now I told that it has the role. tRNA also just now I explained. Then ribosomes, which is considered to be the actual site of protein synthesis. And ATP, just now I told you that ATP is required. Why? It should be energized. That is the energy. ATP is in the form of energy. So amino acids are, and as well as tRNA, they should be energized. So energy should be given. So this ATP is also required. And Mg plus ions also are required for these ribosomes. When they are not involved in the protein synthesis, they are in the two subunits, larger and smaller. But when actual process of protein synthesis starts, these two subunits they join, and for that these mg ions are required. Then different type of enzymes are required. Yesterday also we discussed the DNA or replication as well as the transcription there. All these biochemical reactions are taken place in presence of different types of enzymes. So these enzymes are also very, very important. Then this initiation, elongation, translocation, that is a part of translation. What is that? I will explain you when we will discuss that process. And then after the translation process is over, these factors like ribosomes are released and all these things get separated. These are released. What are these released that we will see? Amino acids form raw material for protein synthesis. As I told you, about 20 different types of amino acids are known to form proteins, and these are available in the cytoplasm. Now you remember that the synthesis of RNA takes place in nucleus, but the site of protein synthesis is cytoplasm because ribosomes are present in the cytoplasm. DNA controls synthesis of proteins having amino acids in specific sequence. Just now I told you. This control is possible through transcription of mRNA. RNAs serve as intermediate molecules between DNA and protein. So that's why at first I explained you the central dogma of protein synthesis. That means the information from DNA to RNA, that is mRNA, and from mRNA to protein. So from mRNA, proteins are to be produced that way. That is why it is considered to be intermediate molecule. Ribosomes serve as site for protein synthesis. Each ribosome consists of large and small subunits. These subunits occur separately in cytoplasm. Only during protein synthesis, these two subunits get associated together due to Mg ions. Means Mg ions help to join these two ribosomal subunits. A ribosome has one binding site for mRNA and three binding sites for tRNA. I will show you in the diagram what is that exactly. And what are these sites? That means for tRNA, peptidyl site, that is P site, amino acid site, that is A site, and E, that is exit site. Now we'll see what are these sites in the diagram, and we'll discuss now actual process, that is the mechanism of translation, also called as synthesis of polypeptide chain. What it involves? It involves three steps: initiation, elongation, and termination. Now, how the initiation starts? Just now I was telling you that for that initiation, what is the main part in the process of polypeptide chain? Amino acids. Now, these amino acids need to be activated. Now, how these are activated? These are activated by utilizing energy from ATP molecule. And this amino acid binds with amino acid binding site of tRNA to form tRNA amino acid complex. Now, I showed you in the video, you saw, observed that how this amino acid gets attached to the 3' end, and in the previous diagram also I showed you AA site, that is amino acid site, that is the 3' end. To that 3' end, This that of tRNA, this amino acid is attached to form tRNA amino acid complex. How this tRNA amino acid complex is formed? First, these amino acids get charged, means they get energy from ATP molecule, and they bind to the tRNA, and that is what called as tRNA amino acid complex. Now this tRNA, which is carrying the activated or charged amino acid, is called as tRNA amino acid complex, and it is now ready for the process of translation. Small subunit of ribosome binds to mRNA at 5' end. Initiator codon AUG is present on mRNA, which initiates the process of protein synthesis. Now see this translation, how it occurs. In this diagram, you can see this is mRNA. It's little bit faint, but you can see all the nucleotides present in sequence. So a group of 3 nucleotide represents a single codon. So now this everything is taking place in the cytoplasm. Now this mRNA has come in the cytoplasm, which is processed mRNA with 5' end and 3' end. Then rRNA, it directs this mRNA to the actual site of protein synthesis, that means the ribosomes. So first of all, this smaller subunit gets attached to this mRNA at 5' end. What is the 5' end? Generally, at 5' end, the process of translation starts with the codon called, which is having AUG nucleotides. The codons with AUG, that is considered to be the start codon. AUG on mRNA. So at 5' end, AUG codon, that is called as initiation codon or initiator for the translation, is present, and to that initiator or that codon, initiation codon or start codon, the activated tRNA amino acid complex, just now I showed you, that tRNA, which is carrying a specific type of amino acid at 3' end, that is AA site, that comes, which are having the complementary nitrogen bases on anticodon loop. Try to understand. Now, this is mRNA with the initiator codon as AUG at 5' end. This smaller subunit of ribosome gets attached to the to the mRNA at 5' end. Then the activated amino acid, which has attached to the tRNA, that tRNA with the anticodon complementary to the codon, that tRNA comes in contact with this, gets attached to the codon. That means AUG is the initiator codon at 5' end. Then what are the complementary nucleotides on anticodon loop of tRNA? UAC. So this tRNA with the anticodons UAC comes and gets attached at the first codon, forming the bonds in between. Then, in this case, after this attachment, the larger subunit of the ribosome comes in contact, and now I will show you what are the 3 sites of tRNA. Now, for mRNA, only one site is there. This is what? 5' end. Now, for tRNA, 3 sites. Which are the 3 sites? Now, where to the first codon, now first tRNA has come in contact carrying the amino acid here at the 3' end AA site. So this is considered to be P site. Then A site is free. Now then what happens to the second codon, that is GGU, suppose. Now for the GGU, the complementary nitrogen bases should be C, C, A on anticodon loop of tRNA. So that second tRNA will come and get attached to the second codon of the mRNA carrying that particular amino acid at its 3' end. Then this side is called right side. This is considered to be AA site of tRNA because from this site amino acids are added with the help of tRNA. So this is called A site. And why this is P site? Because when the second amino acid comes, that second tRNA is coming in contact carrying the second amino acid, then there is formation of the bond that is called as peptide bond. So that is why this is called as P site. And once this peptide bond is formed between the next amino acid and the previous amino acid, now this tRNA becomes free. And this free tRNA is released from this. So that is called as exit of this tRNA. And that's why this site is called as E site. Try to understand. I will show you again in the next diagram. So this is E site, this is P site, and this is A site. Then again what happens? The next tRNA with the anticodon loop complementary to the codons on the third codon on the mRNA, that tRNA will come and then this ribosome will move towards the 3' end as and when this first previous tRNA is released, this ribosome moves step by one codon. Now, once the second tRNA is added, the first tRNA is made free to release, exits, and then this ribosome moves by one codon. When it moves by one codon, A site becomes P site and A site becomes free. So, in this way, one by one, tRNAs with the complementary nucleotides on anticodon loop, they are coming, which are carrying those amino acids, and that peptide bond is formed between the amino acids. I will explain this again and then I will show you. You have just seen the video also. Initiator tRNA binds with the initiation codon AUG by its anticodon UAC through hydrogen bonds. It carries activated amino acid methionine. Always in eukaryotes, it is amino acid called methionine, first one. Or if it is prokaryote, then it is formyl methionine, F-met, this is also represented as. Now, the larger subunit of ribosome joins with the smaller subunit that requires Mg ions. Initiator charged tRNA with activated amino acid methionine occupies the P site of the ribosome and A site is vacant. Now see in this diagram how this. So this is what initiation. Now this is the smaller subunit. This is mRNA. This is the 5' end which is free of mRNA. One more thing you can see that there are few more nucleotides present which are not considered to be the initiator, but these non-coding regions also are present which are important for the smooth synthesis of protein. So here is the smaller subunit first gets attached to the mRNA, then the tRNA with the complementary nitrogen bases on anticodon loop comes in contact with the first codon on mRNA, and then see it is coming in this step. This is the smaller subunit, this is mRNA. Now the first tRNA with the complementary nucleotides on anticodon loop carrying amino acid comes in contact, gets attached to the first codon on mRNA, and this larger subunit binds. Here in this picture you can see here, this is the smaller subunit. Now after adding or of the amino acid or the tRNA, when it comes and with the help of hydrogen bonds, it joins the codons on mRNA. This amino acid is there, which is carried by tRNA. Now this small larger subunits unit binds with the help of Mg ions. Now see, now what happens? This is what called as initiation, because first tRNA when comes in contact, This is what the process has started. That is initiation with the help of AUG. Now, when this is P site, the site is considered to be P site because, and this which is free is called as A site because from this site again next tRNA carrying second amino acid will be coming. So this is called as A site because there is addition of amino acids by tRNA which it is carrying. So this is called as A site. And as there will be formation of peptide bond in between the second and the first amino acid, that is called as P site. And once this bond is formed, this first tRNA, role of first tRNA, previous tRNA is over, so it will exit from the place. That is why this is called as E site. So tRNA is having three sites: P site, A site, and E site. Now see here, second tRNA comes in contact with the second codon on mRNA, carrying the amino acid and having this tRNA with the complementary nitrogen bases on the anticodon loop. Then by hydrogen bonds, it gets attached to the codon on mRNA. And that time what happens? This first amino acid gets attached to the second amino acid, forming the peptide bond in between. And once this peptide bond is formed and these two amino acids get attached to each other, the role of first tRNA is over. So it gets exit. There is exit of this tRNA. It gets released. And so this is E site. Then again, that what will happen? This ribosome will move by one codon ahead towards 3' end. From 5' end, there is a movement of the ribosome towards 3' end, and in this way, one by one, tRNAs are carrying amino acids are added. See, this is the third tRNA. Then again, there is the formation of peptide bond between the second and the third amino acid. In this way, there is a formation of long chain of amino acids, and once the chain of amino acid is formed, these two subunits, large and small subunit, gets released from each other. They get dissociated from each other, as well as these tRNA also immediately, once they release the amino acid, they also are free and they are also dissociated. They also get released in the cytoplasm. So releasing factor, that is called as. And when one by one this tRNAs are carried and get attached carrying the amino acid, that is what formation of the peptide bonds forming the chain, and that is what called as elongation of polypeptide chain. So during this process, activated amino acids are added one by one to first amino acid, methionine. Addition of amino acid occurs in three steps, that is codon recognition, amino acid tRNA molecule enters ribosome at A site, anticodon binds with the codon by hydrogen bonds. The first amino acid tRNA molecule moves to the P site, and second amino acid tRNA is attached to the next codon on mRNA at A site, and these two amino acids join by peptide bond. Here, enzyme ribozyme acts as a catalyst. At this time, first tRNA at P site is kicked off at exit end, that is E site. That is what elongation. And as the ribosome is moved by one codon, this is what translocation. That means the tRNA at A site carrying a dipeptide chain, that is the di means two peptide molecules are there, so peptide bonds are there. So dipeptide at A site moves to the P site. This process is called translocation. Very simple to remember. Trans means change, location is changed. So what is that? A site becomes P site. This is what location is changed. What is that location change? A tRNA at A site carrying a dipeptide at A site moves to P site. This process is called translocation. In translocation, both the subunits move along in relation to tRNA and mRNA. So that's why I told you there is a movement of these subunits, that means ribosomes, from 5 prime to 3 prime end by 1 codon. So once the tRNA, previous tRNA gets released out, that it moves by again one more codon. So in this way the movement occurs, and that is what called as translocation. So the translocation means there is a movement of tRNA, a movement of ribosomes in coordination or in relation with the tRNA and mRNA. During this process, first uncharged tRNA is discharged from E site, and this process is repeated as amino acids are added to polypeptide. So termination and release of polypeptide takes place. Termination means when this synthesis that site doesn't recognize any of the codon. So at the end of mRNA, there is a stop codon. What are these stop codons? Either AUAA, UAG, or UGA. Out of these, any of the codons, if it is there, that means it doesn't recognize any of the amino acid. So that means the reaction is stopped, there is termination of the reaction, and there is no further formation of polypeptide. So this is what called as, these are the termination codons. And what is the start codon or initiation codon? AUG. And UAA, UAG, and UGA, these are the termination codons. And it is exposed at the A site. It is not read and joined by anticodon of any tRNA. So any tRNA will not come because it doesn't recognize any anticodon. So that's why they are stop codon or nonsense codon or termination codons. So this is the last diagram where you can see here. This is a long chain of polypeptide, that is a bond between the two amino acid is called as peptide bond, and as many peptide bonds are present, so this is called as polypeptide chain, and that is nothing but a chain of amino acids. So once it is completed at the last, that means this will not recognize the last codon, this will not recognize any of that anticodon or T on tRNA. So this is what called considered to be stop codon, which terminates the reaction, that is termination codon. And this is what A site, this is the A site, this is the P site, this is the E site, exit site. So exit site, where from where these tRNA when are released from this site. So that is what exit site. Once the amino acid is attached from that tRNA to the next amino acid, then this is free. The previous, now here you can see the tRNA is free. So this gets released. It gets dissociated. It first is gets exit from this site. So it is E site. So in this way, then once there is formation of long chain of amino acid, then these two molecules, that means the ribosomal subunits, larger as well as smaller, they get dissociated from each other. mRNA also gets released. Everything, the amino acid also chain, that chain, polypeptide chain also is released, and which forms in the later stage the tertiary structure which forms the protein. So this is what the translation part of protein synthesis, which includes initiation at 5 prime end of mRNA by the addition of first amino acid at AUG initiator on mRNA, and when again another amino acid is added with the help of adapter tRNA, then this addition of amino acids with the help of tRNA, which forms a chain of polypeptide, that is many peptide bonds between the amino acids, that is called as elongation. and at the end, which do not recognize any of the anticodon on tRNA, where that is called as stop codon on mRNA, termination codon on mRNA, which terminates the reaction of protein synthesis, is called as termination. So the release factor binds to the stop codon, thereby terminating the translation process. The polypeptide is now released in the cytoplasm. Two subunits of ribosome dissociate, and last tRNA is set free in the cytoplasm. That blue colored I showed you. This is last tRNA, not this one. This is a stop codon. That last tRNA, yes, that gets released from the cytoplasm. mRNA also has some additional sequences. See what I was telling at first. mRNA also has some additional sequences that are not translated, and these are referred as untranslated regions. So at first also some first, see, this is what not translated region, which is not involved. This part of mRNA is not involved. This region of mRNA is not involved in the translation. So this is untranslated region. Even the last part of at 3 prime end, this is the 5 prime end of mRNA, which is not involved in translation, as well as 3 prime part of mRNA, which is not involved in the translation, that is called as UTR, that is untranslated region. And the UTRs are present at both 5 prime end, that means before start codon, and at 3 prime end after stop codon. They are required for efficient translation process. I hope you have understood this part. The next part, I'm not going to take it right now, but I'll come to the. So, have you understood? Yes. Okay. Uh, just a minute. I will just keep the. By the time I keep the second video also ready, just if you have any doubts, you note it down there. I'll keep the second video also uh, ready for you. Stop my I stopped my presentation now.